Welcome to Unit 2, Cell Structure and Function. This one's going to be much longer than the first video, I think, just because there are so many topics in this unit for cell structure and function. But I'm going to try to go through them as quickly as possible because I think for most of the topics within the cell things, most of the stuff you probably remember from ninth grade bio, or if you took it in 10th grade maybe. Not much is different between that and AP Bio. There's a few specific things that might be different, and I'm going to try to touch more on those than the minor things. So let's get right into it, I guess. Topic 2.1 is the first one, cell structure, subcellular components. Well, first I want to look at the three types of bacterial cells. We have the spherical shaped in the left, the top left. That is called coccus shaped or cocci. Coccus, coccus. Underneath it is rod shaped or bacillus. And to the right of the bacteria of those three, you have spirilla, so spiral shaped. Now, where you might see a question about this might possibly be something like it might ask if you have a question about say you have an infection that in your lungs that is streptococcal pneumoniae, right? The bacteria that can lead to pneumonia, streptococcal pneumoniae. Well, based on that name, it has coccal in there, streptococcal. And so it might ask what type of bacteria is it? And you can say, oh, it's the circular shaped bacteria. It's not the spirillum, it's not the bacillus type. And yeah. Now also in bacteria you have free-floating DNA. There is no nucleus, no chromosomes, just a big chunk of DNA basically. You do have cytoplasm in all cells, so including bacterial cells. You do have ribosomes in all cells because ribosomes are used to read the DNA. So you have those in all cells. Let's see, you have cell walls in bacteria. That is something people tend to forget. I will come back to that much later on in this. We'll come back to the cell wall thing. But just remember, bacteria do have cell walls, and there's different types for bacteria. However, I don't think you have to know all those necessarily for the AP Bio exam. And they all have a cell membrane, if I didn't say that already. So they have cytoplasm, cell membrane, they have DNA, ribosomes, and yeah, I think those are the things that all cells have, even starting with bacteria. Now, if you look at the bottom left bacteria, the bacillus, rod-shaped, I know on here it looks more like a almost rectangular with curved edges, but it has something called a flagellum on the end of it, and the flagellum is the tail. Flagell means whip, and so you've even maybe heard the term self-flagellate that some religions do, where you whip yourself. I don't know if you've seen that. I think it was in the movie The Da Vinci Code as well, maybe. Um, Now, even in this picture, it just shows one of the bacterial cells having a flagellum, but that doesn't mean only bacteria cells have flagellum. There are other types of cells that are non-bacterial that have flagellum. So let's look at the animal and plant cells. So for topic 2.1 in the AP guideline book, it says to cover endoplasmic reticulum. So that is there's two parts to that. There's the rough and the smooth. The rough endoplasmic reticulum is outside of the nucleus and it has ribosomes, which would make sense because ribosomes turn the mRNA into amino acids basically with the combination of rRNA and tRNA coming in. They all kind of work together to make these amino acid lines to start forming a protein. So right outside the nucleus, it would make sense that the RNA can just come out, the mRNA can come out, and the ribosomes are there. Now the smooth endoplasmic reticulum does not have ribosomes, and that's what makes it smooth. Smooth endoplasmic reticulum. The functions include detoxification and lipid synthesis. So parts of cell membranes, lipids, for example, might be made here. And detoxification, such as breaking down alcohol. So you could have a question asking about if someone who takes some drugs or alcohol, what part of the cell might help in that? That would be the smooth endoplasmic reticulum. The Golgi apparatus, which in these pictures look like pancake stacks, sort of. The Golgi apparatus um, has to do with synthesizing proteins as well and packaging the proteins for trafficking. Mitochondria is mentioned in topic 2.1 as well. So mitochondria, um, they have a double membrane. The outer membrane is smooth, but the inner membrane is highly convoluted, forming folds. You have lysosomes. Lysosomes are little sacs that have enzymes that break things down, called hydrolytic enzymes. And if you remember from the previous video, hydro, water, lytic, break down, lysis, break down. So the lysosomes, which lyse means to break down, lysosomes have things to break, uh, just say cellular debris or cellular waste can break it down. You have vacuoles. Now in animal cells, you have temporary vacuoles. You have small vacuoles that just hold water for little specific reasons, and the water might go in and out of the cell. But in the plant cell, you have the large permanent vacuole. And it has the large permanent vacuole has many purposes such as nutrient storage. Now, chloroplasts are something also mentioned in the book for 2.1. So chloroplasts are found in photosynthetic algae and plants, and they also have a double membrane, um, just like mitochondria do. And we'll come to that at the end of this video, actually. So let's move on. So here we go. Topic 2.2, cell structure and function. So some more things about this one really just talks about the same as the previous one because it wants you to go over organelles as well. Endoplasmic reticulum provides mechanical support, carries out protein synthesis, and plays a role in intracellular transport. So intracellular means within the cell, extracellular is outside the cell. Now also in 2.2, it wants to go over um, chloroplast. So chloroplasts have thylakoids and stroma and things like that. So the thylakoids are stacks of called grana. So a granum is a stack of thylakoids. And the thylakoids is actually, it's almost like an organelle within this organelle. And that is what does the photosynthesis part. Now you have membranes of mitochondria contain chlorophyll, pigments, and electron transport proteins to make the photosystems, which we'll get to that in a different topic when we get to energetics. We'll focus more on that. So you have the light-dependent photosynthesis reactions in the grana. You have the stroma is the fluid with the inner chloroplast membrane and outside of the thylakoid. And the carbon fixation reaction of photosynthesis occurs in the stroma. This is also called the Calvin cycle. Mitochondria, which are actually much smaller than chloroplast. Mitochondria does the Krebs cycle, which is also known as the citric acid cycle, or I think there's even another name. And this happens in the matrix of the mitochondria. Mitochondria also are responsible for electron transport and ATP synthesis, which happens in the inner mitochondrial membrane. So these are important to generating energy in eukaryotic cells. Topic 2.3, cell size. If you look at our pictures there, you have cell A and cell B. Cell A is much smaller than cell B, so cell A, it takes less time for molecules to go through the membrane and to get to where they need to be, basically, compared to cell B. Cell B is much larger, so it takes more time to get to the membrane. If you look at the bottom, the little chart there talks about the diameter of these spheres. It gives you surface area and it gives you volume. One of the important things as far as cell size goes is the surface area to volume ratio. So if you look at the first small sphere of those three, it has a 3 centimeter squared surface area and a 0.5 centimeter volume. If you were to make a ratio of surface area to volume, you would have 6:1, and it gradually gets smaller as you go to the larger cell. That surface area to volume ratio is smaller. You want a larger surface area to volume ratio for a more effective cell. So a smaller cell is better to a certain degree. And that is why cells are so small in the first place. Why aren't cells the size of people? Why aren't we just one big cell? Well, because molecules are very tiny. They need to get to specific places. It is easier for an organism to have a trillion different cells that are very tiny in order to work with these molecules than to have one giant body with these molecules trying to get all over the place. Now here's some relevant equations. These are on your equation sheet that you got from the beginning of the year if you lost it, or who knows what you did with it. If you don't have it, you can just Google and find the AP Bio equation sheet, and these are provided to you on the exam usually, but since the exam's online, I would try to make sure you have a printout of the equations, or at least have them written down maybe. That way, when you're taking the exam, you can have them there. I don't imagine they would ask you to calculate anything, considering that the time limit is so small on the modified exam. But those are there in case you need them. Those are your relevant equations, the different volume and surface area equations for spheres, cubes, rectangles, and cylinders. Now, here are some specialized cells. These are things you need to know for some questions you might get, such as a sperm cell. It might talk about why does a sperm cell have such a odd shape to it? It has a nucleus with the head part, the coil of mitochondria, and the tail. Well, the sperm does not need within the cell body, the head part. It doesn't need anything in there. It's not synthesizing any new proteins. It's not uh, creating lipids. It's not detoxifying. In sperm, all you need is the DNA. As far as a mature sperm, it just needs to get to an egg cell, an oocyte, for fertilization. And the coil of mitochondria, that's a crap load of energy to get that tail to kick pretty quickly. That's pretty much it. And you need the tail for, propeller movement. So this is a specialized cell, and you can talk about how small sperm are for their efficiency reasons. This is one of those points where that surface area to volume ratio is important, because there is a very large surface area to volume ratio for a sperm cell. Now, a root hair cell from a plant, very similar. It has a pointed shape to it because it is trying to get water molecules through the dirt. If it is a pointed shape, it can kind of break through, almost like stab through the dirt particles a lot easier, or the soil particles. And when it does that, it can break up those particles and you can get some water. It's very efficient for root hairs to be kind of pointed. If they were block shaped, it would be difficult for them to break up the soil particles as much. And nerve cells are very long and skinny, and they have these weird branches to them, the dendrites and axons and everything. They are for communication. So it's good to have long cells like that for long distance communication, to go from your brain all the way down to your arm to make your hand move, to make your fingers move. It's very important to have these long cells that can do this type of communication very quickly. That's why the axon bodies are so thin, because all they do is send a nerve impulse, just a transmission of what the message is. The message might be move your finger, and it shouldn't take you forever to do that. It should be very quick. So these have very that part of the cell has a very high surface area to volume ratio, so that the message can move along very quickly. All right, plasma membranes, topic 2.4. Now this is where it can start getting kind of long. So we want to look at phospholipids first. Phospholipids have a hydrophilic and a hydrophobic region. If you can't tell what that is, you can see the picture in the middle. There's two pictures of little cups with a water cup. And if you look at the top picture, you have the tail portions sticking out into the air and the head portions sticking in with the water. If you remember, water is a polar molecule, and the head of a phospholipid is also polar, meaning that those parts will interact with each other. The tail parts are lipid. The tail parts are not hydrophilic, they are hydrophobic. And if you look at our phospholipid on the left, this is the chemical structure. Here's a replacement picture where you can see I've circled the hydrophilic head, the phosphate group, and the hydrophobic tail. And there are two long chains there, and most of the time we depict a phospholipid with that general shape. Oops, didn't mean to make that second one come up. Oh well. But if you look out to the far left, you can see a little head with two little squiggly tails. So that is the general shape. Now, if you look at the second beaker picture where it says water and oil, if you take a droplet of oil and stick it into water, you will see that the tail portions will stick inwards of oil, will stick inwards, and the head portions will stick outwards. Now, this is important because if you look at the bottom picture that I made come up, the bottom middle picture, you have where you can form a shape. The first shape, the second shape could possibly form as well, but what you typically end up having with lipids will be the third picture, which, if you noticed, looks almost exactly the same as a phospholipid bilayer. So cells do have this bilayer because you'll have tail portions of a layer want to touch the tail portions of another layer, while the head portions will stick out to interact with water. So this is important as far as evolution goes as well. There's a good video by Stated Clearly. I think it is it's the one of the evidence for evolution videos, I think, or maybe it's the chemical evolution, I believe, where he actually shows a really good animation of a cell membrane, how it could form in water, and they've actually proven in labs that they can do this in water. Now, out to the top right, we have different membrane proteins that can stick in or out. So you have a surface protein, and it just sits right on the surface. It can form as a signaling type of protein or something like that. And then you have embedded proteins that stick through the cell membrane completely, and these can allow things to go in and out, or it might have some other function. And right below that, the chemical that looks like a lipid, one of our examples from the previous video, you have a cholesterol molecule. It's got a bunch of rings to it and then some tails sticking out. Cholesterol is a lot more thicker than, maybe not thicker is not the right word, but it's not as flexible as a phospholipid. So cholesterol sometimes will be embedded in the cell membranes, the plasma membranes, in order to make the membranes a little bit more thicker. So that is why sometimes you hear doctors talk about good cholesterol and bad cholesterol. Good cholesterol helps out with cell membranes more than what the quote-unquote bad cholesterol does. All right, let's move forward. Membrane permeability. So the membrane of a cell is called a selectively permeable membrane. You can look at this picture if you want to pause on your own time and see all the possible components. I've already talked about the proteins as far as being a surface protein or a membrane spanning protein, it says here, or embedded protein. One other thing is a carbohydrate chain that it shows here. You do have carbohydrates sticking outside of proteins. You can have glycolipids or glycoproteins. Glyco just means carbohydrate chain, basically. These can act as receptor type things or signaling things. Um, but yeah, you don't need to know way too much about that for this topic. So if you look at the chart on the bottom left, you have ions in the type 1 box where it says ions such as potassium, sodium, calcium, and chloride. These all have a charge to them. They are polar. These do not easily pass through the membrane. So you do have a polar region on the outside of the membrane, but because the inside part of your bilayer is nonpolar, these these ions do not like to pass through very well. Glucose is another one. It says it is polar and it's very large. Even if it wasn't polar, it being very large is typically an issue because the membrane usually doesn't have enough space for large molecules to just walk right in. You usually have to have something to help them in, such as a membrane spanning protein. Type 3 molecules, polar and small. These polar small molecules might have some difficulty going in and out, but also sometimes not. Water is small enough and it kind of works well enough with the membrane. Water can go in and out of the membrane fairly easily, but not always. That is why sometimes you have membrane proteins called aquaporins. And it's like a pore in your skin, and the word aqua, water pore, basically. Aquaporin is the protein. And then the last part of the chart, type 4 molecules, it says nonpolar and small. So this would be like oxygen and carbon dioxide. A lot of the times these are easier to pass through because you need oxygen to go into the cell to interact with mitochondria and the waste product of carbon dioxide from respiration to leave the cell membrane. So these ones can move a little bit easier in and out. And then going to the cell wall, I mentioned this earlier. I know the cell wall is not mentioned in any of these pictures, but what are they made up of? Well, for plants, mostly cellulose. There's also, I think, lignin. And then for fungi, you have chitin. Some people say chitin. I think it's supposed to be pronounced chitin. And then for bacteria, peptidoglycan. All right. So then membrane transport, topic 2.6. So this topic focuses on passive transport, and there are different parts of passive transport that we'll get to in later topics as well. But for now, let's just look at simple diffusion. Well, if you notice, let's say the triangle, let's say that is a glucose molecule of some sort or a sugar molecule. Well, if you notice on one side of the membrane, it has all of the triangles, the other side of the membrane does not. Sugar molecules do not easily pass into the membrane. However, if these small dots, if those are water molecules, they can usually work their way through the membranes, aquaporins or other things that let them slide in pretty easily. So simple diffusion is going from a high concentration to a low concentration. Things like to spread out naturally. They don't like to be all bunched up together. It doesn't make sense. So there's a chart that actually on the right side of this that lays out each of the different things for active transport and passive transport. And under passive, you even have the subsections of diffusion and facilitated diffusion, which facilitated diffusion is the next topic, so I will talk more on that in a moment. But let's see, let's go through the chart real quick. Active transport requires energy. It moves molecules against a concentration gradient, so active transport does put a bunch of molecules, it does bunch them up basically. Active transport always involves a channel spanning protein, so you do need a protein to do active transport. Active transport can move ions such as sodium and potassium. There's even what's called the sodium-potassium pump. I recommend you look that up if you have no clue what that is. And also active transport can move large molecules such as sugars. Under passive transport for diffusion, there is a X on it moves the molecules along a concentration gradient, meaning it's going from a high concentration down to a low concentration. Things are evening out a bit. Diffusion, molecules pass between the phospholipids. And for diffusion, it moves small, nonpolar, and polar molecules. Now, it can move some polar molecules that are small, but not all of them. So, again, water is a polar molecule that can move through most of the time. So it really depends on the environment of the cell. So now, facilitated diffusion, molecules move along a concentration gradient. It involves a membrane-spanning protein, also known as an embedded protein. Facilitated diffusion can move ions such as sodium and potassium, and also it can move large molecules. So let's move on. Facilitated diffusion, not a whole lot here. Not a whole lot at all. Just this one picture. Now, if you notice, it says glucose right there. And then you have a hormone. This is looking at a large protein. There's something called a gated channel. It's if we're looking at the first picture of the protein, the step one, if you will. So we have the triangles, which are glucose, the circles, which are hormones. You have a hormone binding site and a gated channel. It's called a gated channel because it works like a gate, like at your house, or maybe around the school or something. Gate it's kind of locked. It needs to be opened before you can go through. Now, even though it doesn't explicitly say it, this hormone is probably insulin because, it's trying to let glucose go into a cell, probably. So then, picture 2 of the protein. After the hormone has binded to the binding site, says channel begins to open. And then for the third picture, you can see glucose molecules that are going through the protein, supposedly going into the cell. So that's pretty much it for this one. Yep, I was just trying to look at the book real quick. Topic 2.8, tonicity and osmoregulation. So osmosis is a form of passive transport, specifically with water. So if you see something about osmo, it means water automatically. If you look at the picture on the top right, it says movement of water in and out of the cells. If you notice, you have a bunch of water molecules in the cell and a bunch of sugar molecules, what it says is the solute. So water is a solvent, sugar molecules are solute, and you have them going in or out of the cell. Now, things like to follow a concentration gradient, meaning they like to be even as far as a percentage goes. That doesn't mean if you have 10 water molecules outside and 5 inside that the outside is going to necessarily go into the inside. It's really going to go to where the solute is. It's going to try to make a balance of the solute inside and outside of the cell. So you want to have like a 5% concentration of the solute inside and a 5% concentration of the solute outside. Well, let's look at the osmosis in plant and animal cells. So you can have a hypertonic, isotonic, or hypotonic solution. Hypertonic is, for an animal cell, is called being crenated. And this is where water is leaving the cell. If it is hypertonic, that means there is a high amount of solute outside the cell. So the tonic is related to the amount of solute outside the cell. Isotonic means it's the same concentration in and out. That is typically where you want your cells to be. And then you have hypotonic, where there is more solute inside the cell than outside the cell. If there's more solute inside the cell, that means more water is going to go inside the cell. And this causes these cells to swell up and burst, and it says it's called lysed. Now looking at the plant cell, if there is a increase of solute outside the cell, the water will rush out, and it is called plasmolyzed. It shrinks up. These are your dead, dry leaves. Now, sometimes, even though leaves might appear to be dead or dried up, as we say, um, there is an extent to where you can actually give water to the plant and you can save the plant. So that might be something that comes up, saying, if you notice some leaves look plasmolized, does it mean that they are dead necessarily? No, not yet, at least not entirely yet. Normal for the plant cells is the E picture in the middle, and the F picture says turgid, and there is something called turgid pressure. So if there is a lot of water, then you are going to have the vacuole swell up, and luckily for plant cells, they do have a cell wall which keeps the cell from bursting, unlike our cells. If you look at the hypotonic for animal cells, picture C, the cell will burst. If you look at picture F for the plant cell for the hypotonic, the cell has not burst yet. However, it can get too much water. I'm sure you guys, one or two of you, have done this before where you've had a plant and you've killed the plant by watering it too much. That does happen. Now, I don't know if you remember the beginning of the school year, we did the potato cell lab, or potato chunk lab, I should say, where we took potatoes and we figured out how much concentration of sugar potatoes have by sticking it in different sugar concentrations of water. So here is something that we did calculate with that lab. However, I'm sure most people forgot it. Luckily for you, this is easy to calculate. This is all in your equation sheet as well. I think there's even more. I think it gives some of the variables, or the pressure constant is there. Never mind. Okay. I think it does give you the same stuff exactly in your equation sheet then. So water potential and solute potential are two things to look at you could be asked to solve. I doubt again that they will ask you to fill out any math. They might, but they might ask about, say they'll say you might take a chunk of an apple and stick it in some water, and then you notice that um, the apple might have shrunk up a little bit. So it would say, what was the type of solution that you put the apple in? You would say a hypertonic. There was more solute outside of the apple than was in the apple, so the apple shrunk up. That is actually very highly unlikely. Apples have a lot of sugar, but I guess that's beside the point. One more thing I wanted to point out, I just made that picture appear on the right side. This is a plasmodium, and it is a protist cell. It is a single-celled eukaryotic cell. It does have a nucleus. Now, if you look, one of the things that's labeled says contractile vacuole. So it has what's similar to a vacuole like the plants, except this purposely tries to expel water. The reason for that is because these cells live in um, water, basically. They live in water environments. They live in watery environments. But so they have to continuously pump water out, otherwise the cell could explode. That's basically it. Let's move on. All right, we're getting close to the end. So topic 2.9, mechanisms of transport, and topic 2.10, compartmentalization. So in mechanisms of transport, this is where it talks about in the book um, active transport. Active transport is almost the exact same as facilitated diffusion, except it takes a ATP binding site and an ATP molecule, which I will cover ATP in more detail when I do the energetics unit. But if you look there at the picture at the active transport, so you might have a substance that needs to be sent somewhere. So let's look at of that picture. There's a 1, 2, and 3 basically. So you'll have a ion binding site, you'll have a ATP binding site, and right now on the bottom of the picture you have a bunch of molecules already over there. So most of the time you'd say, oh, no other molecules need to go over there. In some situations in your cells they do. So you'll have an ATP bond for the second picture, and for the third picture, the ATP will lose a phosphate group and become a ADP. ATP stands for adenosine triphosphate, ADP, adenosine diphosphate. So you're going from 3 to 2, tri to di, meaning that you are losing a phosphate group, and you are pushing those other molecules across this membrane to a already concentrated part. So that is active transport, requires energy, and it's going against the gradient. For topic 2.10, compartmentalization, talks about exo and endocytosis. Exo means exit, endo means to bring in. So exocytosis, you're having these, your temporary vacuoles, as I was saying previously for animal cells at the beginning of this video, saying we had temporary vacuoles, or lysosomes, or other things like that, where waste and debris, or just molecules that need to go to the surface, can be sent out of a cell. Endocytosis brings things into a cell, and there's actually 3 different types of endocytosis. Phagocytosis is basically what's equated to a cell eating. It's taking in larger solid molecules. Pinocytosis is taking in water usually. Sometimes it's called cell drinking. And then receptor-mediated endocytosis. You might have something that binds to a receptor and is brought in that way. And the last topic of Unit 2, topic 2.11, origins of cell compartmentalization. So this started with this is what they call the endosymbiotic theory, or sometimes endosymbiosis, or something like that. Basically, you have a cell membrane and you have digestive enzymes inside of a cell. However, if a bacteria goes into a cell, endocytosis, it's brought in, but you don't have the proper things to break it down, it's possible that that bacteria can be incorporated into something functional within the cell, and that is what they believe happened in early cells. And that is how we developed from all bacterial cells, all prokaryotic type cells, to generating a nucleus, and then later on generating animal cells by bringing in aerobic bacteria, and then even from there plant cells and other protist cells, bringing in photosynthetic bacterium. Now, I do want to point out fungi. A lot of people confuse fungi with plants like mushrooms. They are not mushrooms are not plants at all. They do not have photosynthetic um, components to them. Technically, fungi are more related to us than they are to plants, even though they look the same as plants. However, if you look at very primitive animals, such as a sea sponge, or an anemone or something like that, well, it kind of does look like a underwater fungi of a sort. So I think that's pretty much it for topic 2.